I would now like to, I have the pleasure to present you Dr. Christophe Guinet from the Centre d'études biologiques des Gizeh from the University of La Rochelle, France. And I thank him for his availability and acceptance of our invitation to be here today. The title of his presentation is Studying the Environment and Ecology of Deep Diving Elephant Seals, 20 Years of Census Development. Please, Christophe, the floor is yours.
Okay, thank you very much, Luis, first of all, for the invitation, and thank you for all the people to join. So the title of that presentation could also have been Are elephant seals becoming the white mice of the ocean? And I will explain you why. So as you know, the ocean is warming, but we need to precise where it is warming and how quickly the ocean is warming. And more importantly, as an ecologist, it is really critical to understand what are the ecological consequences of such warming, and from phytoplankton to apex marine predators. And in this presentation, I will show you how oceanographic data collected by southern elephant seals equipped with a broad range of biologgers may help us to address these questions while also addressing the question of the foraging ecology of these marine predators. So, as I said, the Earth is warming, and this is becoming increasingly important over the last century, and I should say over the last decades. And in that context, it is critical to study the ocean, because the ocean are taking up 93% of the heat content of the Earth. So the excess of heat is mainly stored in the ocean. But and to observe the ocean, we can use a broad range of systems, and the most common system are the Argo floats, which are floats which can go down to 1,000 meters, and every 10 days they will go down to 2,000 meters and go back to the surface, monitoring temperature and salinity and transmitting their data through Iridium or Argos. But nevertheless, there are areas where we are lacking information. Those areas are the marginal seas and the shelf regions, the deep ocean, deeper than 2,000 meters, and the polar regions because of the presence of sea ice. And if we look where the warming is currently taking place, we can see that most of the warming is taking place in the polar region, mostly in the Arctic, but also in the Antarctic. And when we look at the distribution of the oceanographic data obtained either by Argo floats here or by oceanographic vessels, we can see that we still have a lack of information and data for polar regions, which are warming the most rapidly. And in that context, this is particularly important for the Southern Ocean. If we look at the heat content, we can see that most of the warming anomalies observed in the ocean are taking place in the Southern Ocean. It is indicated here, with most of the heat content increasing between 30 and 60 degrees south. We also see a major change in wind stress, with wind increasing in the Southern Ocean, and more particularly in the Indian sector of the Southern Ocean. There is a change in the oceanographic properties also, with an increase of the stratification of the ocean and the deepening of the mixed layer depth, mostly taking place in the Southern Ocean. And associated to those changes, there is a change in chlorophyll A concentration, which has been increasing over the last two decades. So to investigate all these change, we can use biochemistry profilers. We can rely on oceanographic vessels, but also on animal samplers. And I will mostly talk about that in that presentation. And we can relate also this change to the long-term population trends that we are monitoring for islands located in the Southern Ocean. So here I will be talking about the work we are doing on southern elephant seals that we are equipping with a broad range of satellite tags. And among them, one of the most important one is the tag, the CTD tags, which have been designed by the Sea Mammal Research Unit, and which they can transmit real-time information about the depth, the temperature, the salinity. Some of these tags can also monitor fluorescence to assess phytoplankton concentration and also dissolve oxygen. But when we are transmitting data real time, we can only transmit 2 to 4 profiles a day. However, if we are able to recover the tags, we can recover the archive data on board the tags, and with information about depth, dive pressure at 1 Earth, temperature, salinity, fluorescence only 4 times a day, dissolved oxygen, but also light and acceleration at a very high sampling rate. But we have to deal with some constraint about the size of the equipment, the fact that we have a limited energy supply on board, working in a cold environment. These tags have to also to sustain high pressure. And so therefore we need to optimize data acquisition and processing and transfer. So going back to the elephant seal, this is a 6-month dive record of a young male elephant seal. And as you can see, this male was diving continuously for 6 months, between 500 meters up to 1,500 meters. And when I'm saying continuously, if we zoom in for 24 hours of the life of that seal, we can see that these seals did only one thing, was to dive continuously, going down to 600 meters and going back to the surface to breathe every 20 minutes. So on average, those seals are diving to 500 meters with a maximum of 2,000 meters, with a mean diving duration about 21 minutes, and a surface, and between dives, they will spend 2 to 3 minutes at the surface. And not only these animals are diving continuously, but they range over extended areas. So on this map, you will see the tracks of 452 seals that we equipped on Kerguelen Island. In red, the female tracks, and in blue, the male tracks. So those seals travel up to 16,000 kilometers during one foraging trip in the ocean. And so by working on Kerguelen Island, we monitor the whole Indian sector of the Southern Ocean, extending from South Africa to Australia. And if we are working in different locations, such as Kerguelen, Marion and Prince Edward, South Georgia, the Antarctic Peninsula, Macquarie Island, we can get a global coverage of the Southern Ocean. And so currently, southern elephant seals are contributing to 80% of the oceanographic profiles obtained south of 60 degrees south, and 98% of oceanographic profiles within the Antarctic sea ice. And thank you to Fabien Roquet and Baptiste Picard to processing all these oceanographic data and to make them available to the whole oceanographic community. So, as I said, I mean, basically, we are monitoring temperature and salinity. So here you can see a track of the seals living from Kerguelen Island, going to Kerguelen, and monitoring temperature. So the redder, the warmer, the bluer, the colder. So these seals went to Antarctica, where he met very cold waters, came back in winter to Kerguelen Island, and you can see the seasonal change between the temperature at the surface in late summer and what you will observe in winter when the seal is heading back to Kerguelen Island. But if we recover the tags, as I said, we can monitor very high resolution temperature, such as here, data, and salinity data indicated below. And if we zoom in for 3-day periods. I can show you the details of oceanographic information we can obtain through these animals through extended periods. And currently, the elephant seals are the only means available to collect high-resolution oceanographic data within the Southern Ocean. And when we look at all the data being collected by seals in polar regions, so indicated in blue, we are, you can see that we have complemented the other observatory systems such as Argo floats and oceanographic vessels. So by including them, we get a better coverage of the global ocean. So on the top of temperature and salinity data, we are monitoring biochemistry information such as oxygen concentration, and these seals left from Kerguelen, went to Antarctica and come back to Kerguelen. So you can see the oxygen records through the whole trip of that seal. We also measure some information about fluorescence, which is giving us some information about phytoplankton concentration. So this is a transect from Kerguelen to Antarctica, and you can see the change in chlorophyll A concentration leaving from Kerguelen and arriving to Antarctica with an abrupt change here by the end of the summer when sea ice is forming. But more importantly, in the last few years, we get increasingly interested in what I will call the mid-trophic level. So between zooplankton to the apex marine predators prey, which are mainly mesopelagic fish and squids. And to do that, we have been using a broad range of new biologgers that I will detail. So one of them is the D-tag designed by Mark Johnson and Pauline. And these D-tags allow us to monitor sounds, but also to monitor acceleration, magnetometry, and to provide GPS location. So with acceleration, we can detect what we call head movement associated with what we call prey catch attempt, which are indicated by these black dots here. So you can see that the seal is going down during the day, going back closer to the surface at night. So you can see this change of the prey vertical distribution through the day. You can also see, because you relate these prey catch events are superposed to the temperature, and so in red, warmer waters, in blue, colder waters, and you can see that when the seals reach warmer waters, prey tend to be deeper. And those prey are for southern elephant seal females, mostly what we call lanternfish or myctophids and squids. And if we look along the track where those preys are located, so here you can see the track of the seals, and in color, you will see the number of prey catch attempt per day. So when it is white, it's less prey. When it is red, this seal is catching more prey. And you can see very clearly when we superpose that to the temperature field, that when the seal was foraging along an eddy, the prey catch rate doubled. And when the seal left the eddy, the prey catch rate decreased significantly. So we are now able to locate favorable foraging areas in relation to the oceanographic context, not only at the surface, but also because we get the temperature and the salinity we can, within the water column, we can also locate those prey at depth. And what we can see is that when the seal was along this eddy, the prey catch rate was much higher, and the variability in the depth range used by the seal was much smaller. So the prey were concentrated in a narrower water layer. And when the seal left, you can see that the prey catch rate decreased and the prey range, vertical distribution was wider. So not only this logger has an accelerometer, but it also has a very important sensor, which is a very high sensitivity light logger. And we deploy that to investigate bioluminescence. So what we have to know about bioluminescence is that 70% of marine organisms are bioluminescent. So bioluminescence is not the exception, but it is a generality in the marine environment. And among bioluminescent organisms, there are items which are likely to be praised, but items which are not likely to be praised. So one of the questions was, are the prey catch attempts performed by elephant seals related to bioluminescent events? So here you can see a dive profiles with the seals going down to nearly 600 meters. These are the jerk determined by the acceleration corresponding to prey catch attempt. And these are the bioluminescence events. And here you can see very clearly that the seals were, most of the prey catch attempt were related to a bioluminescent event. It's not always the case. And so if we look here, we have the seals leaving Kerguelen Island and going westward and foraging along an eddy. And here you can see the change in prey catch attempt per day. So that seals, the prey catch attempt doubled. And here it is the number, the location of the prey catch attempt within the water column along that trip. So if we looked at the inward part of the trip, it's when the prey catch attempt was lower, about 500 prey catch attempt per day. But when these seals reached the eddy, the prey catch attempt nearly doubled. Not only the prey catch attempt doubled, but if we looked where we had bioluminescence here, or not bioluminescence associated with prey catch attempt, we can see that during the inward part of the trip, most of the prey catch attempts were not associated with bioluminescence events. But when the seal was foraging on the eddy, most of the prey catch attempts were associated with bioluminescence events, telling us that it was very likely a different type of prey which was targeted, or at least a different physiological state of the prey. So the latest thing I wanted to mention in that presentation is the amazing creation of Mark and Pauline, which is a microsonar. So what it is, in fact, we had an echolocation system to the seals, which allow us to insonify particles in front of the seals, up to 10 meters in front of the seals, and to try to characterize them and to perform echo soundings directly from the seals. So here, some of the first results regarding the prey-seal interactions. So this is the depth in meters and the time in seconds. So in that case, you can see that the seals was descending regularly. You got a target approaching the head of the seal. So the head of the seal is there. So this is about 5 meters. That's what we looked at using the microsonar. And from the acceleration, you can see that there was no acceleration signal. So no, this target was not a prey. Here you can see a change in the diving depth with the seals slowing down its descent. You can see a clear target, in fact, multiple targets, and you can see a clear acceleration signal showing us that when the target got close to the head, the seal caught something. And here, another case where the seals went up, you can detect a target and a strong acceleration signal suggesting that this was also a prey located by the microsonar. And so as part of Mathilde's work, she was able to show that seals were targeting schooling prey, so multiple targets. And when they were targeting those kind of prey, seals were behaving in a certain ways to catch them, performing a complete looping, swimming on a belly and then upside down before catching the prey. But when they were targeting isolated prey, they would keep traveling horizontally. So just looking at the behavior of the seals, the swimming behavior of the seal, you get some ideas of if the seals are targeting schooling prey or isolated prey. And as part of Martin Tournier's work, so using the microsonar, we look at the microsonar data, but through the whole foraging trip of the seals. So this is the records where the seals departed from the coast. And this is the records obtained over several days. So the microsonar was working one day and half the second days. And if we zoom in on a 24-hour period here, you can, you can very clearly see from the microsonar what I will call nictemeral migration of an acoustic layers going back closer to the surface at night and deepening during the day. But that layer was not the layer targeted by the seals to forage, because if you look at the prey catch attempt, they were located much deeper during the day and a bit less deep at night. But this layer could be the prey of the prey. And also because we got the temperature records at the same time, we can relate the vertical distribution of those layers to the oceanographic context and to the vertical structure, temperature structuration of the water column. And if we zoom in, so we have one female equipped with a microsonar from Kerguelen, and this is the foraging trip of that females, and in yellow, the part where we got the microsonar data. And if we look here, this is the temperature field. The black dots, as always, the prey catch attempt. And here you can see the number of echoes per ping produced by the microsonar. So give you an idea of the biomass of organisms present within the water column. We don't know what they are, but you can tell there are more or less of these organisms, depending where you are. With here, for instance, a lot of detections in comparison, for instance, to that area. And also, you get some information about what we call echo strength. So you have also some information about the size of the echo and how reflective is that echoes. And this is a work that Clément and Jade have been doing. First of all, they try to quantify the number of echoes according to its size. And you can see that most of the detections are about very small echoes, which are about 3 millimeters. And as the size is increasing, the number of echoes are decreasing. And if we log transform this distribution, we can see that. I mean, that we got very few large echoes. Not only we can quantify the number of echoes according to their size, but we can also look at the acoustic intensity of these echoes. So as you can see here, with echoes ranging from 1 to approximately 10 pixels, the acoustic strength is increasing. But interestingly, for larger echo size, you can see a bimodal distribution within the acoustic intensity, which is likely to tell us that those organisms are likely to be different between the high intensity and the low intensity organism. or we can see that here very clearly for larger type of organisms. But at the end, we still don't know what those echoes are. And so one idea is to complement that the information we are getting from the microsonar from a micro camera. There were cameras, video cameras deployed on elephant seals, northern elephant seals by Japanese colleagues, such as Naito-san. And this is really nice to identify elephant seal prey. But unfortunately, it's not precise enough to target small, to identify small targets. So we are working on a new micro camera with a high resolution, to get high resolution picture to characterize small targets, even targets which are not used or foraged on by southern elephant seals, but mostly to characterize the biological environment sampled by those southern elephant seals. And the idea at the end is really to combine all the information we have on the prey catch event associated in red with bioluminescence, or in black without bioluminescence. And with the acceleration, we can also characterize if the prey catch events were taking more time. So like here, you got bigger dots, or maybe less time, so smaller dots, likely to be indicative that the prey targeted might be different during the day here or at night, but also with bioluminescence information. So you can see that, for instance, we got a very clear bioluminescence layer, which is not associated with prey catch events. So that bioluminescence is produced by organisms not targeted by southern elephant seals. And in association with the data provided by the microsonars, as well as the behavioral information we have from the seals' foraging behavior when they are approaching the prey, to know if they are targeting isolated prey or schooling prey. So at the end, what we are really interested in is how change in physical properties will affect phytoplankton biomass and composition, and what will be the consequences on the composition and quality of what we call the mid-trophic or intermediate trophic levels, ranging from copepods to mesopelagic fish and squid. And ultimately, all this influence the foraging success of southern elephant seals. So to conclude, I would say that over the last 20 years, southern elephant seals have become increasingly useful in monitoring the Southern Ocean, from physical to biological data. And they are allowed to collect that information at a fine temporal and spatial scale. And I think they provide unique information to address questions at the interface between physical biogeochemicals and biological oceanography. And if you have any question, I will be happy to address them. So thank you very much for your attention.
Thank you very much, Christophe, for this amazing talk. It's really fantastic to see how technology has evolved and how it enables us as scientists to collect multiple data and multiple variables that will help us understand foraging behavior and to integrate that with environmental variables and characteristics that will help us understand better the ecology of the sea. And thank you for sharing this amazing work that you guys are doing. I will give now a minute for people to pose their questions and to vote on the questions which are already there before I give the floor to Christophe to answer them.
So in the meantime, what I may say, I mean, It's not the work of Christophe Guinet. It's really a teamwork. I mean, associated many researchers and students over a long period of time. And this will not have been achievable just by one person.
Of course, this is definitely a team or a multiple team effort to bring together all this knowledge and to develop these approaches. So I guess we have enough questions and we have already clear votes. So I will start with the most voted question, which has jumped out of the screen now. Okay, here we are. It's from Amanda. Do you think this biosensor can be used for analyzing the interactions between other predators, such as cetaceans and other seals species, I guess?
I think, I mean, I'm not really sure. I mean, between what she means between seals and whales, but yes, this sensor in any case can be deployed on other species. I mean, I should tell it's probably easier on seals because what I should tell is to recover those data, you need to recover the logger. So if you don't recover the logger, you don't have any data. And so this is much easier to be performed on seals than it is to be performed on whales, unless you got suction cups and flotations.
Thank you very much. Next questions. Do the tags harm the skin of the seals or affect the animals in any other way?
So one thing I should have precise is for the microsonars, the sound characteristics or the frequencies were chosen not to interfere with the hearing of the seals. And of course, when you add on some loggers on the seals, you are inferring with the hydrodynamism of the seal underwater. So, but we were very careful to assess what is the impact of all this equipment we are putting on these seals on their foraging performances and demographic performances. And this was done by our Australian colleagues, where they have been able to compare the demographic performances in terms of foraging success, winning mass, survival of females equipped in comparison with non-equipped females. And they could not find any differences between the two groups. This is not to tell that this equipment doesn't have any effect on the seals, but it's very likely that the seals are being able to compensate for that. And also, this is probably true for elephant seals because, in a way, the way they are foraging is more like fruit picking than active chasing of the prey. If you compare, for instance, like you will observe in the fur seals or in the king penguins. So it's not a very high energy hunt of a prey, but it's mostly you're moving slowly through the water column and picking your prey. And in fact, what we have been able to show with the microsonar is that most of the time the prey did not detect the elephant seal approaching, or they detect the seal at the last moment.
Thank you very much. The next question, are prey capture events happening more during a descent or ascent phase of the seal dive?
Neither during the descent or the ascent, but during the bottom phase of the dive. It's really the bottom phase of the dive is the foraging period of the seals. They do catch some prey when they are descending and ascending, but really they concentrate their foraging activity at the bottom. And the depth of the at the bottom depth is indicative of the location of the upper part of the prey layer.
Really fascinating. And now, the next question, can the microsonar discern fish from squid?
We don't know. We don't know because as I tell, I mean, we got just the size, the acoustic reflectance, but we don't know if the fish have a different acoustic reflectance of a squid. So we need to perform experimental work under, I would say, control experiment, putting squids and fish, and to see if the acoustic target strength is different. But we are hoping to address that question with the micro camera combined with the microsonar.
Mhm. Fantastic. And now for the final question from Sophie Odell. What would you need in terms of Argos telemetry performance to improve your research field?
I don't think, I mean, the main constraint with Argos system is message size. And so we did a lot of efforts, I mean, to process on board most of the data. I'm talking about pressure data and acceleration data. So to get, for instance, not each pre-catch attempt, but to get, for instance, a summary of the number of pre-catch attempts performed during the descent, during the bottom of the dive, and during the ascent. But at the end, what is really limiting is the message length. So I don't think it will be possible to do everything we are doing currently when we are recovering with the equipment, trying to transmit all that information through Argos. I mean, we are trying to improve the quantity of information we are transmitting through Argos, but we will still be highly limited by, as I said, by the Argos message length.
Thank you so much, Christophe. So we're getting to the end of this session. I would like very much on behalf of the European Cetacean Society to thank again the presenters, Christophe, Jacob, and Michael, for their fantastic lectures and for sharing their knowledge, and to all of you who came and joined us at this event. We invite you all to be tomorrow on the afternoon for the next session of the ECS webinar, which will have another group of interesting, fantastic lectures. So thank you very much, and it was a pleasure to have you all with us.
Okay, thank you. Thank you for having me.